So look, thank you everyone for coming along. This is a really great turnout. So thank you for A, coming, and B, being very punctual. And we have people from all around Australia, and we also have people from over the pond as well, from New Zealand. My name's Stephen Manos. I am the Associate Director of Cyber Infrastructure from the Australian BioCommons, and I'm responsible for all things computing and infrastructure and data storage and various other services for life sciences and bioinformatics. So, and it's really great to be here and have a community of people who are interested in and care deeply about compute and methods and how it all intermixes with bioinformatics and life sciences research. I just wanted to start off with an acknowledgement to say I'd like to acknowledge the Whadjuk people of Noongar and pay my respects to the elders past and present. And I'd also like to pay my respects to the elders whose lands you're all on today across Australia and across New Zealand as well. Before we get started, I thought it'd be good to just spend a few minutes, and if I could ask everyone to just think about the idea of community. There's many of us here who don't necessarily know each other. And I think it's important when we get together in a community like this, especially for the first time, that we establish some ground rules for how we engage and how we talk as a community. So I just wanted to get maybe 3 or 4 words from all of you collectively. Anything that comes up, any words you can think of that you can relate to when you think of a welcoming community. So anyone can just perk up and say a word. Just take yourself off mute.
Sharing.
Thank you, Tiff.
Friendly.
Thank you.
Safety.
Great.
Inclusive.
Thank you. Anyone else for one more?
Respect.
Excellent. Thank you. That's great. Look, I think we can all agree that we want to share ideas. We want to be friendly to one another. We want to make this a safe space for us all. We want to be as inclusive as possible, and of course, we want to be respectful to one another. So, thank you very much for everyone for sharing, and I'd like everyone to adopt those phrases and those words as we go into this next hour and a half. And for future meetings as well, as this is the inaugural and first of many, I imagine. So I'd encourage you all to keep yourselves on mute if you're not talking. Please use the chat extensively. If you've got any commentary or questions, please just type them in the chat as we go. I would also encourage you to use the Zoom thumbs up or hand clap feature as well. Seems a bit dorky at times, but it's actually very effective without making too much noise. So please use those features. And if you do ask a question in the chat, I will be monitoring the chat as well as others. So we'll make sure we kind of come back to you and respond to your question. And of course, I'd encourage the speakers as well, if they do see a comment or a question in the chat, to use that back channel to respond. So just a quick overview of this community of practice and a quick introduction from me. So with the support of the Australian BioCommons and ABACBS, the Pawsey Supercomputing Centre established a bioinformatics community of practice, and this is its inaugural meeting. The expectations of the group really were drafted as follows, and I'd like to actually have some time at the end of this meetup to kind of revisit these and think through what people also want to do. Is really was around, an idea where we could get together nationally, support collaboration, share research projects and opportunities and exchange ideas, discuss emerging technologies, tools, workflows and pipelines, provide a safe environment to support our, collective knowledge and growth in this area, and also gain insights into what we're all doing. Today is really the first community meeting where we're going to discuss some new technologies and activities in Australia for you to all hear about. And you're going to hear from experts from Pawsey, our hosts, from the Australian BioCommons, from ABACBS and from COMBINE. So I will stop sharing my screen And I will give Sarah a 10-second warning and to say that our first speaker of the day is Sarah Beecroft. Sarah is a HPC Research Fellow at Pawsey, who specialises in bioinformatics pipelines and infrastructure, and she's here to give us a high-level overview of the Pawsey Supercomputing Centre.
Great, thanks Steve. Thanks for the introduction and kicking off the meeting. So yeah, we're very excited to be hosting this today. So this will just be a few minutes, just a very high level overview of Pawsey before we give you a little bit more information about what we've been up to and what we're planning to do in the future. Could you go to the next slide, Audrey? Yeah, keep going. So Pawsey is one of 2 Tier 1 HPC facilities in Australia, and there's a subsection of the team pictured here. We're about 60 people, thereabouts. So we're funded through the Pawsey University Partners, which at the moment is UWA, ECU, Murdoch and Curtin, but also the state and federal governments. And we'll be looking to maybe change up or include more partners in the future. So we provide expert support across a lot of scientific domains. So the origin of Pawsey was actually from radio astronomy, but we have grown into supporting things across definitely biology, chemistry, and other domains like computational fluid dynamics as well. So there's a lot going on here. Next one. So there's 2 key systems. Nimbus is our cloud service, and you'll hear a lot more about that later from my colleague Audrey. Nimbus has about 3,700 CPU cores. We also host some GPU nodes. So there is a total of 58 terabytes of RAM. So with the cloud service, as you might already sort of expect, you spin up your own instances or virtual machines. And so then you have your choice of compute specifications, software environment, and operating system. So you're in total control over your VM, and you can do whatever you like with it, except mining Bitcoin. Please do not do something like that. When I say whatever you like, for doing your science specifically. And on the supercomputing side of things, we're actually in the middle of upgrading to our new system, Photonics. So Photonics is the Latin name for Quokka, which, as you know, I'm sure, lives on the island of Rottnest off the coast of Western Australia. So this will be replacing our 2 previous supercomputers, and it is going to be much, much bigger. So it's going to have over 200,000 AMD CPU cores, also more than 700 AMD GPU cores, and 500 plus terabytes of RAM. So this will be the fastest public research supercomputer in the Southern Hemisphere when it is complete, to our knowledge. So we're very excited about that. And there's a lot of work going on in the background to get that working. We've also updated our storage systems. So Acacia will be our 60 petabyte S3 object storage system. And so this is exciting because you can access it from anywhere. If you're familiar with S3 object storage, it provides a lot of flexibility. And so this is more for the life of your project. So while you're running things on Photonics, you can access Acacia to push and pull your data. Banksia will be our 70 petabyte tape storage. And so that's for longer term, slower access sort of data storage. Yeah. So next we'll be talking about how Pawsey is interfacing with bioinformatics specifically. So I think I hand over to you, Audrey, here, and start off talking about Nimbus.
Thanks, Sarah. I'm not sure if this is the next on the agenda.
Okay, sure.
That's great. We can get into that detail later on, Audrey,
okay.
when we go into the detail. So thank you very much for that high-level overview, Sarah. So next we're going to move. So look, if you've got any general questions for Sarah, just please type them in the chat, but we'll keep moving for the time being. So next I'd like to introduce Jeff Christensen. He's the Associate Director of Engagement and Operations at the Australian BioCommons, and he's here to give us an overview of the Australian BioCommons. So thank you, Jeff.
Thanks, Stephen. you can see the screen. Great. So as Stephen said, I'm the Associate Director of Engagements and Operations. That's community and research engagements at the Australian BioCommons. And if you've not heard of us, this is what our mission basically is. We aim to support life science research communities with community scale digital infrastructure and to enable access to computational services that provide obviously analysis capabilities and also to provide training and support solutions that enable researchers to adopt these types of approaches. We are primarily, our core funding is through Bioplatforms Australia, which is federally government funded through the NCRIS program. And we also, there is a large amount of investment, co-investment from a large number of partners, which I will discuss shortly. So I just wanted to highlight, this is a community of practice meeting, and one of our focus is definitely to focus on community scale, whole of communities and community scale infrastructure. So I thought it would be useful to just reflect on the size of the community and the diversity of life science research community in Australia. So these are some figures which we came up with a few years ago. So there's about 100,000 PIs that hold publicly funded research grants in Australia, and about a third of those are in the life science area. So that's about 30,000. And we estimate there's another additional 200,000 students. So it's quite a large number of researchers we're talking about. The spread across sort of, I guess, human and health and medical to agricultural to environmental and conservation is shown here. So about 50% in health and medical, 30% in ag, and 20% in conservation. There is a broad range of experience when it comes to informatics. So by far, the largest group in this life science areas, we would still regard as being sort of wet lab researchers who might be occasional users of bioinformatics services or tools. So they might do a blast online or something like that. And then we sort of move into people where, or groups where they, they may have some access to some sequencing data for the first time and are, trying to make sense of that data. So, omics data is a critical contributor to their research outcomes. So that group is still fairly large. We estimate around 35% of researchers. Then we move more into, I guess, groups where the research is more or less fully dependent on bioinformatics, like every day. So this group is smaller, maybe 10%. And this could be, groups that, do reanalysis of human genomics data in cancer research, for instance. And then we move to a sort of smaller group, but very specialist, where the research is pretty much exclusively focused on, technique, algorithm, or tool development. And we classify this group as bioinformaticians, but really, I guess you could classify anyone in this group across all of these are actually bioinformaticians because they're applying bioinformatics techniques to their biology research. So that's a large and a very diverse group of researchers in Australia, and how we try to engage with this large group is to engage through broad topic areas. So we generally, identify a sort of, I guess, common informatics methodology and form a community around that. So some examples are shown here on the slide. So genome assembly, annotation, microbiome analysis, et cetera, You see here that I put command line users in the yellow. And I think that is an example of this group we are currently, that we are. And I guess our aim is really to facilitate interactions between those groups of life science researchers and a whole range of computational providers to build a more fit for purpose, flexible and cohesive data analysis ecosystem. So this is across institutions, across national facilities, and also internationally. And we work with a lot of partner organizations. You'll see here that, national, like Pawsey and NCI, ARNet are all on here. There's also state-based organizations such as QCIF that run a Queensland-based HPC systems and cloud. We work a lot with increasingly with commercial providers such as Azure and Amazon, also with institutions. You can see here Sydney and Melbourne are core partners. And on the bottom row there, you might see Elixir, Workflow Hub, BioTools, BioContainers. These are all examples of a large international collaboration that's in Europe, and their aim is to, I guess, build coordinated infrastructure across many European countries. We are dealing with those. Sorry, can you see just the one slide or the notes? Because they're not my notes.
Just the one slide.
Excellent, good, thank you. That would have been embarrassing. So I thought I'd just highlight some examples of our work so far, and I've just sort of picked out activities where Pawsey have been a key partner, and one of these communities we're engaging with and delivering services for is genome annotation. And so we actually had a launch earlier today for a hosted Apollo service. So if you don't know what Apollo is, it's a GUI-based system where people can upload their genome assemblies, annotation files, and evidence files, and really go in there and do some manual curations of the automated annotations. So that is the Apollo portal is the service has been developed by QCIF, and that's all sitting on infrastructure that's provided by Pawsey. And you can see that it's we've been working with a number of early adopters, so there's already 11 groups who are using that across Australia, and it's open to anyone. So it's actually launched today. If you're interested in genome annotation, get in touch with us or visit that site. I also wanted to point out, so Audrey has been working with us to set up a system where we can provide fully subsidized access to a commercial genome annotation pipeline. It's called FGenes H++. Effectively, we are covering the license, and we have a license deal with the vendor, and we can have I think 12 users per year in our current licensing setup, and we might change that over time if it becomes more popular. I just wanted to point out that again, 2 of our early adopters have found it really useful. So it's been used by a group in the University of Sydney to generate a high quality genome annotation for our marsupial, and also by a group in Southern Cross University to generate one for T-tree. And so those have been recently published, those papers. I also wanted to point out that Pawsey also effectively supports a wide variety of other types of research areas, so genome assembly, microbiome analysis, proteomics, metabolomics, and also COVID-19 researchers. And they do this by providing significant computation that sits under Galaxy Australia. So if you don't know what Galaxy Australia is, it's a graphical user interface. You just get on, it's a whole system to do bioinformatics analysis in. It contains over, I think, about 900 tools. It has about 400 reference data sets, and it's extremely well resourced. Thanks to some new infrastructure that's just gone in, it's capable of, for instance, assembling a plant genome in less than 2 days. So it's pretty significant. So I guess this is more of a HPC command line user group. So I just wanted to point out some things that we're also doing in this space. And I guess our work here is really about trying to point towards this more unified ecosystem. So we've developed something called Tool Finder, and that really provides a national view of bioinformatics tools and containers, and also where they're installed on various infrastructures, including Pawsey, NCI, on Galaxy Australia, and also on the Queensland infrastructure there. It also has links directly off to these international repositories, such as biocontainers that I was talking about. So if people are interested in finding where containers are, it's all there. So it's like a one-stop shop. A lot of work has also started around optimizing bioinformatics tools for specific infrastructures. So here are some tools. You might recognize some of them. And our teams have been working to get these tools working efficiently on the infrastructure. This is an example of Canoe it's actually on NCI, but the same type of work is happening on Pawsey. You can see here that, you know, the assembly time for this particular thing went from about more than 2 weeks to about 3 days on Guardi. And that was achieved by using a wrapper script that makes the distributed jobs from Canoe compatible with the scheduler on Guardi. So that's an example of the type of work we do. I also wanted to point out that it's not just tools, but also workflows. And this is something we're increasingly interested in describing and being able to, I guess, get to a point where we, you know, workflows are more reusable on more types of infrastructures. So there's another tool called Workflow Finder, and I'll send out all the links to these afterwards. And this is an example of this one's actually Sarah has been doing the documentation for this particular Cromwell implementation of a GATK variant calling pipeline. And so this is just showing the very top, but it's a very well documented pipeline there. I also wanted to point out that we have a pretty vibrant training program, lots and lots of events. And one of our most popular, I guess, workshops has been around using containers in bioinformatics, and that was a joint effort between Pawsey and us. It's happened twice. I'm sure it'll happen many more times. It's very, very popular. And then I saw some names from people from the University of Sydney on the attendee list. And a really nice example of how we've been working with the University of Sydney is to, the team there has developed some very nice webinars around really, you know, well, what do you do when you're outgrowing your institutional compute? Where do you go next? And also a webinar about, you know, well, if you get to the point of having to submit an NCMAS application, how can you do, you know, your best job at submitting that? Just wanted to point out that most of all of the webinars are recorded. They're available on our YouTube channel. You can go view them anytime. Yeah, I've probably spoken long enough. So I guess my take-home message is, you know, really how we operate is it's a conversation. We're always very happy to hear what the community needs and for us to then act as, I guess, that lens between the community and the infrastructure providers. So I just wanted to say there's lots of ways to get in touch with me. I'm in the Slack channel, Jeff Christensen. You can email me, visit our website, or tweet us. So thanks very much.
Thank you, Jeff. What's the best way, I guess, in terms of people knowing about events and hearing about what's happening? What's the best way to do that? I know you just put up that slide with those links. Is that available somewhere, or can we distribute it somehow?
Well, we also have a newsletter, so I'll put a link in the chat to subscribe to the newsletter because it's monthly and we advertise all of the events coming up each month.
Yeah. Okay. Are there any questions at this stage for Sarah and or for Jeff? Anyone? All right. Well, let's keep going. So we're going to break up the talks with just a few interactive, I guess, sessions, short, kind of 10-minute Q&A sessions, and they're going to be run by Aditi Subramanya, who is the Marketing and Events Officer at Pawsey. So I'll hand over to Aditi to host this for the next 10 minutes.
Hello, everyone. Just on the note about sharing links and everything, we will collect all of this. This event is being recorded as well. It will be uploaded onto Pawsey's YouTube channel. And before I upload it, I'll try and collect as many links as I can to the presentations, newsletters, etc., and also include that in the video description. But until then, I'm just going to share my screen and we'll go into a quick poll. Okay, so if you go to menti.com and type in the code, I'm not sure if you can see it. Yep, if you go to menti.com and use the code 53705700, we have a quick poll just to understand how often you use computational resources other than your own laptop or desktops. Those are things like the cloud, high performance computing services, other services, not necessarily Pawsey or NCI or the kinds of examples that we've given today. And this will kind of help us for future events for this community of practice to get a bit of an understanding of the computational users and also to help you access those resources in the future, depending on which stage of your research you're on as well. Later on, my colleague Pascal is actually going to give a really nice overview of how to kind of recognize when you will kind of evolve from using your laptop or your desktop, your personal machine, and step into cloud services or high performance computing services as well. Nice, it's a really good spread. Of course, the poll is nice and vague, so it's really open to interpretation on this one. Those of you that have answered the poll, this is your opportunity for a quick break before we get into a couple more presentations. We'll have another break as well with another poll, but if you want to make yourself a cup of tea or do anything you need, we'll probably get back to chatting at about past, so in about 2 minutes.
Sounds good. I wanted to ask a question. Is anyone heavily engaged with their institutional HPC provider? So at a university, I guess a departmental or university level HPC cluster? And Rob, would you be willing to quickly talk about that for a minute?
Yeah, I'm happy to. Hi, everybody. I'm Rob Edwards. I'm at Flinders, and we have a pretty reasonable HPC cluster. It's mixed use, but I would say the use is dominated by bioinformatics applications. Everything else, there is some physical chemistry stuff happening on it, but who knows what they're doing. What in particular, do you have anything in particular you'd want to know? I mean, that's
actually great. I just wanted to get an overview of where it was. Do you have a good experience there? Is it challenging, or is it generally
no,
smooth sailing?
we have a good experience. It's it's something that's been improving. There's a lot of history of issues in SA, but it's something that we've been working on to improve and enhance. Storage is always an issue, of course, but we have a few hundred terabytes now of storage that we use, and so it's something that's been developing over the last couple of years. Yep.
Thank you for putting your hand up for that, Rob. Much appreciated. And maybe one more. Ido, apologies if I pronounce your name incorrectly. Would you be willing to say something for a few minutes?
Yeah, thanks. Happy to. Yeah, my name is Ido. I'm from Griffith Uni. And yeah, we have here a local cluster that's called Gowanda. And it just got a sort of a lift up makeover recently, I think last year. And it's all right. It's for a sort of a small to medium size HPC. So it's definitely not as big as the national ones. But it provides some flexibility. So sometimes there's really downtime and there's not a lot of people working on it, and it's great. So you have the resources available there, and you can get to the head of the queue very quickly. Sometimes a lot of, I wouldn't give names, but I would say a lot of physics people sort of jam up the queue, and it's really hard to, and they don't manage their jobs properly, to my opinion. So yeah, but it just gives another option, which is great. And another thing which is good, that you've got a more intimate relationship with the admins. So I know my admin really well. I can give him a call anytime if there's any issue. So that's the advantage of having sort of a local HPC cluster.
Great, thank you. And maybe Ma, can we hear from you for a couple of minutes?
Sure. Can you hear me there? Yeah.
Yeah. Hi.
I'm at the University of Melbourne. So we have an HPC system called Spartan. And I think it's pretty, I was just trying to see if I could find the specs quickly, but I can't. But I guess the specific thing I wanted to say that's maybe a little bit different to others that have spoken. I'm in the Melbourne Data Analytics platform, which is a centrally funded platform that was established to kind of help researchers enable data intensive research. So we play kind of an intermediate role between the infrastructure providers at the university and the researchers themselves. So I've been sitting inside the HPC team for half a day every week. Yeah, and so that's been a really, really good experience in terms of bringing researchers' concerns to the HPC team and then being able to communicate things from the HPC team that they want researchers to know in a way that's more accessible to researchers because we're researchers ourselves.
That's great to hear. Thank you. Nice to meet you. And maybe one more from across the pond, Matt. What's your view on Nessie's HPC?
Apologies, sorry, right in the middle of serving up.
Sorry.
Yeah, no, so Denny and I both work for Nessie, and we are more or less providing exactly what is being discussed here. So we work in the support team, and we provide training environments and looking at workflows and all exactly the same things that are going on. And I see some really cool stuff that we're going to be able to take forward and look at and share and support and develop with our users here. Great.
Thank you, Matt. Thank you, everyone, for answering that. That's a really great spread of answers and intel into what sort of resources people are using and so on. So thank you very much. We'll move on to our next speaker, and please keep the questions coming from in the chat as well. So next up, we're going to have Nicola Armstrong. She recently started at Curtin University as an associate professor in data science, and she's a member of the ABACBS executive committee, and she's here to tell us all about ABACBS. Is that how you pronounce it, ABACBS? Yeah. Over to you.
Okay, so can everyone hear me? Yep, and can you see just my slides and not the?
Correct.
Great. So hi, my name's Nicola. I'm an associate professor in data science at Curtin University in Perth, and I'm also a member of the ABACBS executive. And I'm here talking to you today as a member of the ABACBS executive, even though our president, Jimmy Breen, is also on the call, yet he told me he couldn't because he was busy. But I'm also a Pawsey user. So I guess that makes me the person in Perth to talk about ABACBS and Pawsey. Sorry, I just realized I didn't have my video on. So these are Jimmy's slides. But anyway, I'm going to give you a brief overview of what ABACBS is, what it hopes to do, and what it's involved in across Australia. But first up, bioinformaticians are getting into the news. You see here is Torsten, who won one of our inaugural Open Science awards from the association 2 years ago now on news. So we're getting attention. We are getting recognition that we are a proper profession, and we deserve recognition also from the funding agencies, and that's something that we would obviously like to improve all the time. we're also, a lot of what we do involves data science. And so data science is, of course, the sexiest job of the 21st century. And a lot of people think that they can do data science very quickly and easily. But there's a lot of background knowledge that you need to have in terms of statistics, computing, for bioinformatics, biology, that can't just be taught or learnt in a 2-day course. So I think everyone on this call knows that there is a massive amount of data being produced by sequencing centres worldwide and also in Australia. And the pipeline is narrowed extremely when it comes to compute time and compute expertise. And we are essential to getting out the important results as bioinformaticians. We play critical roles in a lot of analyses that are being done in medical research institutes and universities around Australia. So why do we need our own society, ABACBS? Well, it was started because we slipped through the cracks in a lot of places in terms of recognition. There's no department of bioinformatics. It's very hard to find undergraduate degrees in bioinformatics, although they're starting to come back at master's levels in a lot of universities more recently. The requirements for bioinformaticians are pretty specific. It's an interdisciplinary science, which means that the research that we do is hard to categorize, which means also that training programs are scarce. It's hard to find bioinformaticians, and the funding structures suck if you're a bioinformatician, I think is the basic thing. And we're also much less likely to be awarded fellowships because a lot of our influence in publications is not as first or senior author, but we're very critical second or third author, but that doesn't get recognised in the scoring systems for grants. So these were all reasons why there was a push, obviously, to have our own professional society so that we could then hopefully negotiate or advocate for bioinformaticians on a national scale. So this society was started in 2014. It's relatively new through the support of what was then the Australian Bioinformatics Network and other community members. And ABACBS's aims are to strengthen both our science and our profession, to encourage and support students, and to provide representation and advocacy and promote interaction and awareness. A lot of the time, bioinformaticians are on their own in a wet lab, and being able to discuss with another bioinformatician how you analyze data, how you solve a computing problem, how you solve an analysis problem, in the same way that the wet lab guys discuss the best way to extract DNA or how many minutes you got to run the centrifuge on, etc. Those sorts of nuances, to be able to have someone that you can bounce those sorts of things off, to have that sense of community is important to bioinformaticians. So the vision of ABACBS is, of course, for excellence in the science and profession of bioinformatics and computational biology. We really would love to have good career opportunities for bioinformatics and computational biology, for our importance and our contribution to science that is done in medical, biological, agricultural research to be recognised as not just being service providers, that we do actually contribute in the same way that other members of a collaboration might contribute. And for people to understand, I guess, why bioinformatics and computational biology are important. So in order to do that, we run an annual research meeting, which is joint with COMBINE, which is our student arm, and you'll hear more about them later. We have started this year, I think it was this year, a national seminar series, which runs every month on the third Tuesday of the month. Then we aim to have a more experienced bioinformatician and a more junior researcher every month talking about their research. We support local events in terms of the Sydney Bioinformatics Research Symposium when it's able to run, and also the Victorian Cancer Bioinformatics Symposium. And we also support BioC Asia, being the bioconductor platform with R. We host a lot of online training workshops, especially we support them through our COMBINE training arm. And we also support lots of other local events when and if they can run during lockdowns. So we have several official subgroups. COMBINE, as I said, you'll hear about more later. We also have a special interest group or subcommittee of professional bioinformaticians, those who do not work in academic research jobs or teaching and research jobs, but might work in core facilities or industry. We have a special interest group for postdoctoral researchers, particularly emphasising career options and paths for bioinformaticians and other things that might be interesting to the postdocs. And the special interest groups, BioC Asia and the Sydney Bioinformatics Research Symposium. We also have an education and training committee where we're involved in training the next generation of bioinformaticians. We're interested in education, as in at a university level, so official courses and units in bioinformatics across Australia, as well as training, so short training courses for wet labs or for bioinformaticians who want to learn something on the side. And we're, as part of the education and training subcommittee, we're also part of Goblet, which is a global organisation for bioinformatics learning, education and training, and they advocate putting a lot of their training materials or materials that have been used in training courses online for open use by the community. And of course, we're also part of, we support and are part of data carpentry and software carpentry. So one of our successes that we've done in the last few years is we now have a 4-digit code for bioinformatics and computational biology as part of the Australian New Zealand Standard Research Classification. And we're hoping that that will hopefully also flow on potentially to more grant funding if we have our own code, that we won't fall into the cracks of not being computational and not being wet lab. Therefore, we sit in the middle. How are we judged? How are we ranked? So hopefully that will start to improve things as well. We have our national conference will be held in Hobart this year. There will also be local hubs in, I think, all the major capital cities, including Perth. The Perth hub will be at UWA for everyone that's here. Registrations are open until pretty much the conference starts, but abstracts close on Monday. And, oh no, my slide didn't save. I've got one more slide, which I really want to show you. So I'm going to stop this share, but there is a very important event coming on that I promised that I would show. And now I've got to find my slide. Oh, can you tell that it is still teaching in Curtin and I am completely disorganized? But we also support and sponsor or part of AMSI, the Australian Mathematical Sciences Institute, and I particularly co-organised and involved in organising Bioinfo Summer. And Bioinfo Summer will be run this year as a virtual event with local hubs at the University of Adelaide or the South Australian Genomics Centre. I don't know where the physical location will be. There's also going to be one at James Cook University in Queensland, UNSW in Sydney, Curtin in Perth, and of course the University of Melbourne in Melbourne. And one of the themes for this conference or this summer school is high performance computing for bioinformaticians. So if you are one of the newbies, if you like, to high performance computing, I highly recommend that you register for Bioinfo Summer. Most Australian universities are members of AMSI, which means if you're a student, it's only, I think, going to be $90 for the week or something. It's ridiculously cheap. Yeah, so that was all I wanted to say. Thank you.
Thank you so much, Nicola. That was great. I feel like you touched on lots of extremely personal and relevant points to many people on the call. So I'd love to hear some initial thoughts from people, or commentary about being the in-betweener or the et al. Kind of person on the publisher list. So on the author list. So anyone got any feedback or anything to add? Questions to ask?
while we're waiting to see if anyone's got any questions, I can comment on my own use of high performance computing. I guess when I started out, it was my laptop. And then I moved to having a local server at my institute, which I still use quite heavily because as a statistician, I do other analyses, not just bioinformatics, but also I like to play around with my data live. But having gotten next-gen sequencing data, that is when I've moved to using Pawsey in particular and having my raw data stored at Pawsey as well. Because I've just outgrown what I have, but I still do like to use all of the smaller, lower levels of computing as well.
Great. Jimmy, I was really interested in your comment around the for code change. Can you add to that point that you made in the chat at all?
Yeah, I mean, somewhat anecdotal at the moment, but just in terms of the people, the feedback that we've been getting from the community that previously, especially for ARC fellowships, but a little bit less so for NHMRC, but that we are rarely successful in getting those fellowships. And since the sort of the for code change has come in, we've actually been seeing a few sort of examples of sort of senior or mid-career sort of bioinformaticians who are being uptake. It's actually interesting whether this does also coincide with the MRFF combining with the NHMRC to sort of potentially fund a lot of the clinical bioinformatics, which is actually really, really crucial for a lot of molecular tumor board meetings, for example, which, Nicola, I am supposed to be in at the moment. So yeah, anecdotally, I'd love to get some data on this, and we can, and I guess plans to perhaps do a survey to our members, perhaps during our conference at the end of the year.
In my opinion, though, Jimmy, we had a couple of investigators get up this year. I don't think that was more than in previous years.
Yeah, I mean, I've just heard from a few people who have said that they had the feeling that there was a sort of more success this year. So
I think the proof might come more with the ideas.
Yeah, exactly. Yeah.
Yeah.
All right, thank you. And Nicola, there's been a request for the link to register for both events that you described. So if you can put that in the chat, and we can also distribute after the meeting, that'll be great. So thank you again. So we will now move on to a short presentation by AJ and Sushintha. I think it might be co-speaking, both co-chairs of the COMBINE 2021 Symposium, and they're here to tell us all about that. Thank you.
Everyone, just give me 1 second to share my screen. Can you all see that?
Yes.
Fantastic. Hi, everyone. I'm AJ, a PhD student at the ANU, and I'm joined by Sushintha, a PhD student at Deakin University. And today we'll briefly talk about COMBINE. As Nicola just described, COMBINE is the student subcommittee for ABACBS, and we're also the Australia region student group for ISCB. And our vision is to promote education, collaboration, and research for students involved in bioinformatics and computational biology. COMBINE has a lot of programs for Australian research students, and we run a number of workshops throughout the year for free, along with a number of outreach and communication events. And I'll just briefly touch on some of them. We run the annual COMBINE Student Symposium for research students in bioinformatics and computational biology, along with 3-minute thesis style competitions, hackathons, various career forums, and a number of networking and peer mentoring sessions. The COMBINE committee is a really diverse group of people around Australia, from the undergraduate all the way through to the early postdoctoral level. And we think we do a great job of representing students around the country. And I'll briefly just talk about our annual workshops that we run. We run workshops for R, Python, introductions to transcriptomics and scripting in the command line. And finally, I'll just touch on our 2021 Combine Symposium, of which Pawsey is the principal sponsor. So the symposium is held as a hybrid event on the 22nd of November, and it's a fantastic opportunity for students to look at current research, present their research or early ideas, and also establish collaborations and networks of people working in a similar field. And Suchintha will just drop a link to that symposium in the chat. So if you have students who are doing bioinformatics, there's still time for them to submit an abstract. And that is all. We're happy to take any questions if there are any.
Thank you so much. Any questions? And again, feel free to type them in the chat and we can answer as we...
I see one in the chat. Yeah, we do upload our workshops for people who have done them. So all the resources are there and the workshops are recorded. I don't believe they're publicly accessible at this time.
Are they going to be publicly accessible eventually, or?
Yes, we are working on to make them publicly accessible in the future. So yeah. We also have a channel called Ask Anything in Slack, in the Combine Slack. So if you have any questions, you can post on that. And the other students, if they have worked on that specific topic, they will answer it. So that's a very good way to get to know resources and to get to know people.
Thank you, Suchintha. All right, let's move on to our last presentation for the day. I'd like to welcome back the Pawsey team, particularly Audrey and Ali. Sorry, the second last presentation. So Audrey and Ali to give us some more detail around Pawsey and bioinformatics.
Thanks, Stephen. So we'll talk about how Pawsey and bioinformatics talk about how bioinformatics is at play at Pawsey. So firstly, we'll talk about Nimbus, but before I go on, we have got a dedicated team here of supercomputing application specialists like Marco and Sarah, and myself as a bioinformatics systems administrator. And also we've got visualization specialists like Ali and Yathu, and also lots of ongoing training. So we have got a very strong focus on bioinformatics at Pawsey. So let's go on to talk about Nimbus first. It is often users first, I guess, contact with Pawsey. It's quick and easy to sign up and have an allocation, and it's based on command line and also supports graphical user interface. Now, Nimbus has got lots of pre-built images that users will select from as their operating system, and we recommend them to use Ubuntu 20.04 as the image of choice to run as their operating system. Now, coming this quarter, we have got a dedicated bioinformatics image, basically built on top of a Ubuntu operating system. And we will include a range of utility tools that we find bioinformaticians love using, such as Nextflow, Singularity HPC, which I will touch on next, Ansible, Python, and R, and also other, I guess, supporting or dependency type of software. Now, Singularity HPC is a container registry. Basically, it enables users to use containers, but not having to know all the nuances of and syntaxes of running a container. So basically, Singularity HPC was written by came up by one of the Singularity developers. She came up with this registry so that a bunch of different recipes are already in the repo, in the registry, and it's also a community-based type of registry where people can submit pull requests for new recipes to be put on there for different tools. So this is an example here on the left of a list of bioinformatics tools. we use the prefix key.io/biocontainers because that's where most of our bioinformatics tools are can be found. And again, it is a community supported one, so it's got a huge range of different tools that we can find, and basically each of them have the prefix key.io/biocontainers. And to pull the container or to install it, you just have to run shpc install and then the prefix and the tool of choice. They are then used as modules, such as you will on HP systems. So your module load that tool or that container, and then you run the container as though you would be running the tool, as a standalone. So in the back end of things, Singularity HPC does all your syntaxes that's needed. That Singularity, you don't have to type in Singularity exec blah blah blah. You just run fastqc if you're using fastqc. So this module format can be used on Nimbus. And then when you go on to, say, if you also have access to Citonix or HPC, you can still use the same, use your containers in the same way. Now, we also looked at having some automated tool deployment, especially for interactive sessions for RStudio and Jupyter Notebook, basically catering to R users and Python users respectively. And how that is done at Nimbus, or can be done at Nimbus, is based on an Ansible playbook that we have basically developed. So you run this command line and you will be prompted for things like what sort of R version you like. You set an RStudio password, and also you let when prompted, you indicate what your SSH public key location is on your local computer, because that's needed to be able to forward the port, and then for you to open your browser for the interactive session. Basically, when you do that, you're allowing your local access you're basically accessing the Nimbus instance from your local computer and running the interactive session. And all that's done with that one single line command. Same thing for Jupyter Notebook, you can do the same, and you run that command and you basically give the same prompts, answer the same prompts. So, okay. So those are the things that we have been working on and we find will be really useful for a lot of bioinformatics users. And so moving on to the access, the application process is pretty simple. You go to the website, which is apply.pawsey.org.au, and this is all there is on the right-hand side, questions that you have to answer. So we will try and make it as simple as possible, as long as you can prove or show, what you're trying to run, especially things like what tools you're using. You can basically justify what sort of, instances you require, and what sort of storage you require. It's usually approved within a week, and you get an access of 6 months. Before the end of the 6 month period, you will get a notification that it is coming up, and you can renew it using the pre-filled form that you did from the start of the application. And then you renew it for another 6 months. And that's all from Nimbus side. I'll pass on to Sarah, who will talk to us about Citonix.
Thanks, Audrey. Yes, so as I said before, Nimbus is the cloud part, but we're also in the middle of deploying our really big supercomputer. So this is aimed more at the really big computationally demanding jobs that would struggle to run on other infrastructure. This is really good for parallel processing, using MPI, or just using a lot of cores or a lot of nodes. And it will also have that really good GPU capability as well. So that is obviously really useful for machine learning and those types of jobs in particular, but there are also some GPU-enabled, more standard bioinformatics tools that are increasingly becoming available. So at the moment, Magnus is our largest system. And so this runs at 1.1 petaflops. So this is like how many operations per second. Photonics phase 1, which is coming online sort of the middle of next year, will be more than twice, 2.5 times the size of Magnus. But when we get to Photonics phase 2 being on the floor, which will be in 2023, this, as you can see, will be just a ridiculous increase in computing power, which is really exciting. So you can imagine the type of science that you'll be able to get done using this type of facility. So access to Photonics is done through merit allocation. So the process is a little bit more complicated than the one pager that Audrey just demonstrated. But when you have your allocation, you're not bound to using a specific subsection or partition of the computer. You can use large scale or run large scale jobs across most of Photonics. Can I have the next slide, please? So this is just talking a little bit more about the availability timeline. So at the moment, we are open to people that are interested in becoming a Photonics early adopter. So this gives you early access to the system. Also users which put in a merit allocation for 2022 will end up having essentially a lot of free compute available to them as the Photonics phase 1 comes online. So you ask for, say, however many, 100,000, 200,000 core hours. You actually have a lot more available to you next year, essentially for free, which is exciting. We will also have a sort of early adopters phase for Photonics GPU. And then in 2023, then you can access the whole thing. So just to give a bit of a hardware overview for those of you that are into that type of thing. So the phase 1 will provide the CPU compute, the super fast interconnect, which is important for getting a supercomputer to run as one unified system rather than just having, for example, a bunch of laptops. The high memory and visualization nodes will also be available, and Ali will talk a little bit more about the visualization capabilities coming up. We also have a high performance file system, so that reduces any sort of file read-write latency. In phase 2, we'll get the rest of the CPU compute, production level GPU compute, and much faster interconnect speed. And Acacia is in the process of being deployed as well. And so this is that S3 capability that I was talking about earlier, which will really improve the interface of accessing your data over the duration of your project. So in particular, we've been working on bioinformatics features that we know people like, and this is definitely a work in progress, and there's a lot more happening in this space over time. But for example, things that are available now include workflow managers such as Nextflow, SnakeMake, or Cromwell, which really improves the reproducibility of your research, but also introduces like nice checkpointing. So if your job fails, you can just restart it without having to go back to the beginning. We also have a lot of popular bioinformatics tools pre-installed. So these are available as modules. So for example, rather than having to manually install, say, SAM tools, you can go module, load, SAM tools, and it is available to you. So that saves a lot of time. We do also have containers available using the Singularity interface. So you're using a Docker container, but accessing it with the Singularity like Docker engine. And we, as Audrey was talking about, we also have a Singularity HPC registry or Singularity HPC available. There's also pip and conda for doing your own local install. So that's a common misconception that you can't install your own software on HPC. You can do it. It just needs to be for you as a user or your project group. You can't install it for everybody. And we also have a Cromwell GATK4 pipeline, which is currently available. So yeah, there's a lot coming up in this space. Oh, sorry, next slide. So just a word on the allocation schemes for next year. So there's two ways currently to get onto Photonics. So one is NCMAS, or the National Computational Merit Allocation Scheme. So this closes, I think, next week. So this is open to the whole Australian research community. So this is a little bit like applying for a grant that you need to justify or explain your project, your methodology computationally, which you are planning to use, and how much compute you need. There's also the Pawsey Partner Merit Allocation Scheme. So this is only for partner universities or partner institutions. So Curtin, ECU, Murdoch, UWA, and of course CSIRO. So this is open for a little bit longer. Thank you. Yep, so deadlines for our applications. So 5th of October for NCMAS and 11th of October for Pawsey Partner Allocation Scheme. And there is a lot of online training available for these, which I'm sure we can link to in the chat. Next slide, that might be it. And so again, talking a little bit more about the early adopters. So all current Magnus projects will get information about potential to be an early adopter towards the end of this year. And then early GPU call will be in the second half of next year. And there will also be access to the Topaz GPU cluster for GPU migration purposes. So next. Okay, so that brings us to visualization. So Ali will take over from here and talk about what's happening in that space.
Thank you so much. Talking about the visualization, I will give you a very brief kind of high-level introduction to what sort of visualization services is offered at Pawsey. It could be summarized into remote visualization services as well as visualization lab and expertise. So remote visualization service will be in two different systems, will be offered in two different systems, Nebula and RemoteViz. Nebula is Windows-based. That's the boldest difference between both systems, and RemoteViz is Unix-based. So whoever that has access to supercomputer already is a supercomputer user will get automatic access to RemoteViz based on the current workflow. But to access Nebula, they have to apply. It is in early adopter phase at the moment. However, soon that will be announced officially to the research community, and identical application procedure to supercomputer will be introduced and practiced to access Nebula. More detailed difference between two systems is that Nebula is more sophisticated system coming with the latest, almost latest Intel Zone generation, the gold, whereas RemoteViz, which is Topaz based, actually, it comes with the silver version. The number of the cores that you get in Nebula is ridiculously higher than RemoteViz. It comes with higher memory as well, also available in two low and high memory modes. Upon request, you can get access to high or low memory. Both of them are NVIDIA based in terms of the provided graphic. RemoteViz has RTX 5000, whereas Nebula is equipped with the latest RTX 6000, as well as very latest A6000 with 48 gigabyte of the graphic available. It comes with dual GPU as well. Both systems are very similar. They will provide the desktop remotely in the GUI, I mean, and both of them are web-based. Remoteviz.pawsey.org.au, pretty straightforward. If you are supercomputer user, already you have access. Nebula.pawsey.org.au, after applying for the application and accepting, you get access. It is web-based, interactive as well. So you have to book the machine first and then go and use it. Next slide, please. And visualization lab. This is very new service that we are going to announce it in October and officially release it to the community. It is currently available for early adopter as well, same as Nebula. It comes with the micro LED display technique, which is the latest Sony technology. 4K UHD in 120 Hz, stereoscopic enabled, and the brightness and contrast are stunning. It is available to be used with Windows as well as Linux, so it's integrated with Nebula as well as Remoteviz. The researchers, they can bring their own laptop to connect it in 60 Hertz or even like wireless sharing. So they can have video conferencing there as well as VR, MR, and XR will be supported. So I think that's all about the platform and services that we offer. The expertise comes down to setting up the workflow that suits you best to have the most efficient kind of visualization. For example, in situ visualization is what we are going to offer this year, to start to offer. The question that we have from the community is that how often do you visualize your bioinformatics data? What kind of application do you use? We have some researchers from, for example, Peter Mac Cancer Research, that they are working on genome data as well as COVID-related research. What you can see in the question slide, that's a sample of visualized spike protein of the coronavirus. So please share it with us. What sort of requirement do we have? What sort of application do you need? If you send it simply to help@pawsey.org.au, we investigate it and look after installing on Nebula or Remoteviz. Bottom line is that the visualization service at Pawsey is cross-platform with introduction of the Acacia. When Acacia is in production, you can have all of the systems integrated together. You can transfer data between Pawsey file system, Nimbus, as well as Nebula or Remoteviz, and there's no limit. Have fun.
Ali, Audrey, and Sarah, thank you so much for that comprehensive overview of all of the great things happening at Pawsey. I'd ask you if there's any questions, please just pop them in the chat. We're going to keep moving on to our last chat from Pascal Alaki, who's the HPC specialist, or a HPC specialist at the Pawsey Supercomputing Centre. So we look forward to hearing from you, Pascal, on bioinformaticians and their HPC pathways.
Yes. All right. Hello, everybody. Hopefully I am sharing my screen.
Yep.
Okay, there, perfect. So yes, as mentioned, I'm a HPC supercomputing application specialist. Work with Sarah, the same team, and obviously work with Audrey and Ellie. And what I want to do here is just trying to get you thinking about the workflow, because transitioning to a supercomputer, even though some of you may have used larger computing infrastructure, using a supercomputer is not necessarily the most straightforward task. And it does require you to think about how you can make your, achieve your science faster, right? So it's not as simple as just having a bigger laptop. And so what I want to do here is get you to think about that workflow, identifying when you need to actually use a supercomputer, because sometimes you might just need to scale a little bit, not a lot. So we're going to talk about what is an e-research workflow and get you thinking about yours. We're going to do this by doing a cake baking challenge. And so thinking about how you could, what is that workflow for cake baking and how does that actually relate to your workflow and how you might optimize. And eventually I'll just mention how can we help, who can you get help from, and what sort of infrastructure or help you should try to seek considering your workflow. So, I mean, the basic nominal workflow might break up into collecting data, staging some data for processing on the supercomputer, then visualizing the outcomes of that data, and then archiving it for later publications, for later use, etc. Now, that basic idea that that workflow could involve some serious processing at any point along the way, or possibly at no point along the way. So you can collect data, but the collection of data might be, as I said, a physical process. You have a wet lab, you actually are getting data. For other people, it's just pulling data from online resources that exist already. So for a lot of people have been playing with visualizing COVID data. So they technically are not necessarily interested in, and their collection is not involving actual collection of COVID data. They just rely on databases existing that are up to date, and they pull it and then try to do some analysis on it and then visualize it. That may not require lots of computing. It may require lots of computing depending on how complex or complete that data set might be. And then of course you would like to archive it for further use and also for possibly posterity. So how your workflows might go could be something like this. It could be portions of this. Perhaps you're heavily involved in the processing and visualization of data, but not necessarily doing any staging. And the question might be, well, so what bits can I take of my workflow and make efficient use on a supercomputer? Or what bits need to go to a supercomputer? What needs to go beyond my laptop? What would break your laptop? Because the visualization, perhaps you could just run on your laptop, but perhaps you actually need some of the services that Ali mentioned, the Magenta Compute. If you don't have if you are trying to render lots of stuff and you don't have the GPUs to render locally, you might need to move that data somewhere so that you can then visualize it in a three-dimensional fashion. So the idea is to think about this workflow. So I'm going to take an aside, as it were, it seems, and bake a cake. And the reason we're going to bake a cake is what is involved in baking a cake. So if I think about baking a cake and saying I want to produce a cake with icing in 1 hour with one baker, right, just one person baking a cake, what would that workflow look like? So if I think of the full workflow, it might be really complicated. So there's really early preparation that you might need to make a cake, such as growing the chocolate, like the cacao plant, and then harvest the beans to produce the chocolate, then raise some chickens to harvest the eggs, etc. But that's a lot of early preparatory work that I assume cannot be offset to something that is, you can't really speed that up, but it's there. I mean, you might argue that the really early preparation is make a universe from scratch. Then you can have something, then go on and make a cake. But then making the cake, you're mixing flour, typically eggs, flour, oil, milk, chocolates, the ingredients. You mix it up, usually put in a tray, a mold of some sort. You heat the oven, place the tray in, and bake for a little while, and then get a cake out. And that workflow, that coarse grain idea of, I'll just grab some ingredients, mix it in a bowl, pour that into a tray, heat the oven, put the tray in, make my icing separately, wait for the cake to bake, and then put some icing on. That seems fine if you have one cake and you have one baker with one kitchen, and that's the goal, right? That doesn't seem too bad. You don't need to necessarily think about, well, what's going on in each particular step. It can be quite coarse grained. But what happens if you need to change it, right? You need to bake 1,000 cakes in an hour, or a cake in 10 minutes, or an enormous cake, a 1-ton cake. And then it gets a bit tricky. You're like, well, how am I going to do this, right? And the reason we're going with this is just thinking about, like, what would you change in your workflow? So 1,000 cakes in an hour? Well, you could say, I could argue that, well, I just need 1,000 people with 1,000 kitchens kind of near each other, and they just do the cake that takes an hour to bake, right? But it's not that trivial if you have to have something coordinated, right? So if you want 1,000 cakes in an hour, you want to make sure that everybody kind of starts at roughly the same time, so you end up with 1,000 cakes after an hour. And then that in itself makes you think about, well, so how do I coordinate that? Am I asking everybody to synchronize a stopwatch? Do they have all their ingredients set up? Do I have to make sure that they set up the kitchens the same? So the kitchens maybe aren't the same. Maybe the kitchens are the same and it's exactly the same setup, but maybe it isn't. So I need to start thinking about, how do I coordinate this? Someone has to chat and talk to the bakers to start baking now, or also possibly accounting for failures, right? If you have 1,000 people in 1,000 kitchens, but one person burns their cake, you've now not succeeded in your 1,000 cakes in an hour. You've only got 999. There's also the other stuff you might have to think about where you're like, well, if I want to make sure that the cakes are exactly the same, not just 1,000 cakes, but 1,000 of the same cakes with the same icing and the same ingredients, then someone has to go get the ingredients, give it to the bakers to make sure that it's all uniform, and then take the final products and transport them somewhere else. And so that type of workflow means I need to think about, before I just had ingredients, bake a cake. When I was like, well, ingredients, move the ingredients to the kitchens. Are the kitchens big enough? Where are the kitchens? Where are the bakers relative to the kitchens? Then make sure they start all at the same time, bake a cake, and then I got to transport those cakes somewhere. So it ends up being a bit more complex. The 10-minute cake also might be very complex because you need to maybe you can get more people to do each separate tasks. But is it in a large kitchen? Can you have several kitchens? How do you make sure that they do the independent tasks independently and they don't get in each other's way for tasks that depend on each other, or even in the independent tasks, right? If someone's pouring flour in a tiny bowl that you also want to pour the sugar in, then basically it's not possible. You're stuck. You know? So then you need to start thinking about how to coordinate that task so that whatever bit you're doing doesn't interfere with another bit, and that you can do it at the same time. And the tasks that wait, do wait, but they have to communicate. They can't just visually, like you can maybe see if a baker says, oh, they're done pouring, but they might not know. Maybe the person needs to get back and get a second cup and pour some more. So there's a lot of coordination that needs to go on. And that means that you need to think about, well, what goes on in actually making a cake? And maybe in a slightly different fashion. You break up the tasks differently than you would in 1,000 cakes in an hour versus a 10-minute cake. And that, as I said, that role ends up being playing. So now you have your 1-ton cake. Can you translate the how you would break up the tasks and scale them up with more people, possibly bigger kitchens? Do you need a bigger kitchen? This would probably need a bigger kitchen. I couldn't fit this cake in my kitchen. I don't have an enormous kitchen. I just have a regular sized kitchen. But other people might have a very big kitchen if they're running a restaurant. But maybe this is too big. Unless you maybe break it up and stick it all together somewhere else outside the kitchen. So maybe a lot of little cakes stacked on top of each other. And of course, then it gets, if you have the really big workflows, it is effectively the example of, baking 1,000 1-ton cakes in 10 minutes, right? That's the exascale supercomputing enormous problem where you're like, well, that's what I want to aim for. Then you really need to think about some heavy details about how you start doing this, because that is each one is sort of a slightly different angle, but if you need to combine them all, it's a really complicated issue. So I want you to think about your workflows and think about what bits are essentially isolated. They can be done on their own. They can't be sped up. They need some time to do, and that's it. And there's a bunch of tasks that wait. There might be a bottleneck in your pipeline where you have to think, well, I need to do a bunch of stuff. I could do a bunch of stuff in serial, but then the pouring, let's say, of the mixture into a tray, you have one bowl and one tray. Maybe you can't do it any faster than that. You can scrape them with a spoon to kind of speed up the flow, but that's it. So you're there's a bottleneck there. And there's a bottleneck that maybe means that some places you have to really think about, can I do that independently, or somehow can I speed that up? And how does this relate to your workflow? Well, hopefully you've got some ideas of how this might relate to your workflow. And I think the idea of, if for people who have not used supercomputing, then it's a good idea to kind of draw up some workflows. And for even those who have, because we've seen a lot of people at least that have answered the poll, they use computers that are not their desktop or laptop. That might mean that you still kind of do the monolithic approaches. I have a bigger desktop effectively elsewhere that runs a little faster. But that's not a supercomputer. The idea of a supercomputer is parallelism. Can I perform multiple tasks simultaneously? Some tasks might be can't shrink down, but you can do 2 tasks at once. So the idea is to always remember that a modern supercomputer is not simply just a faster processor. Like it's just a faster machine. So it does an answer faster. The example would be maybe that little red bar in that task is a fixed size, and you can't shrink it. but you can do every other task parallel to it. So great, that's how you have to treat a supercomputer. It's not just making, it won't necessarily take a task and shrink the amount of time that's taken to do that task. So there's a bit of things to think about. And that's when Nextflow, Snake makes some of these workflow managers are really useful as well to kind of figure out how you can break up your tasks and do things independently. So how could you optimize your workflow? Is it in the processing phase? Is the visualization something where Magenta has a bunch of GPUs and do portions? Is it in the staging of data that you could maybe use essentially implicitly the parallel file system that exists on supercomputing systems to really kind of get rid of essentially a read and write bottleneck that is the main issue with your workflow? Where could you do this? And what could you benefit? How could you make use the benefits of essentially maybe a faster file system, fast network connections, or many computing elements to your advantage?
Pascal, just quickly, we might need to wrap up in a minute if that's okay. Thank you.
Yeah, so I'm gonna basically kind of end here with this discussion is like, so what do you need, right? So some of the problems end up being that there's insufficient memory on a laptop to run the program, and that could be migrated to maybe supercomputing or Nimbus clouds. The data sets might be too large or just might be too long. So the idea for, and it's kind of been mentioned, is cloud compute, and specifically like Nimbus Pawsey compute, is really designed to kind of provide you moderate speed up requirements. So a few hundred cores, some fast transfer rates. It's kind of easier to manage. You kind of do the management. But if you then need to go beyond that and use thousands of cores, that's when you really want to use a supercomputer. We provide several stages. We've got the supercomputing that can have enormous memory and fast compute. But there is a leg up if in case you're finding that you just want to move to a slightly bigger desktop to kind of figure out how you might use a supercomputer. Nimbus is a good place to start, and then keep going for it and get bigger and bigger compute. And so I'll end there. And if there are any questions, I'm happy to answer.
Pascal, that's great. I found your cake analogy very tasty. So thank you very much for that. I enjoyed it. Much appreciated. And Matt, how did your dinner go earlier?
This is the leftovers. Oh, where's it gone? Where's my screen?
Yeah, I can see it.
Sorry, there's no fried egg.
Looks delicious. Thank you.
I'll just say, really cool set of talks from the New Zealand side of things. Really cool to see all the parallel work that you guys are all trying to do. Very similar to here. Very cool.
Thank you, Matt. Hey, we're kind of at the end of our session, so really big thanks to everyone. We just wanted to run a very quick Menti question while we're chatting and closing off the session. And Aditi, if you could just, if we can post the code maybe in the thing. So if everyone can just go to their browser, and it's really just asking the question of how often do we want to meet up to make this useful. So there's another question of what sort of topics do we want to engage in, whether it's around infrastructure or pipelines or hearing from other facilities that provide computing resources, or even hearing from each other. What are we doing? What are we working on? Maybe a series of lightning talks from various researchers, but we'll follow up, I think, after this talk with a survey to get, just with a few questions to get a bit more information from you on perhaps some of the topics that we can focus on. So if everyone who's here can just get into Menti and just answer that question, that would be a really great way to assert where we want to take this. And also just wanted to say a big thanks to Aditi who set this up and corralled all of us into this event. So thanks very much for your efforts today as well.
Thank you.
Well, it looks like there's a firm statistically relevant result at 3 months in my reading as a physicist. I think it seems like the winner. As I said, we'll be in touch with some further information as we move towards planning that next session. And we'll also follow up with a message with various links and resources that came about from today. So Aditi, any final words from you?
No, I just wanted to thank everyone firstly for coming and attending this first meeting. It's always a little bit scary trying to create a community and bring people together. Didn't want it to be too quiet, but no, this event has been fantastic. We've had a really good range of speakers across our various organizations that are really trying to come together and push the field of bioinformatics from various perspectives. So thank you all for joining us. Please complete the survey that I will email out to you so that we can get some feedback on future catch-ups, whether you want them to be formal or informal, and the topics around them as well. And don't forget to join the Slack channel if you haven't already. Thank you. I think Matt might want to say something. No, he's just got his hand up. Well, he just wants to show us.
No,
he just wants to show us eating his nothing.
No, just a clap.
Just clapping.
Thank you, everyone.
Yeah.
Thanks very much.
Thank
Enjoy
you,
the rest of your
and
day.
thank you, Stephen, for emceeing this event. That was awesome.
Very welcome.
Okay.
See you later. Goodbye.
Hey guys.